Let's move on to an important topic in the physiology section and review the baro and chemoreceptors. Here is the baroreceptor and chemoreceptor diagram from your first aid book, and let's first discuss the locations of these receptors and what they detect. There are two important locations, high-yield locations of these receptors. They exist here in the aortic arch, and they exist here in the carotid sinus. Baroreceptors detect changes in pressure. They respond to arterial wall stretching. Chemoreceptors, on the other hand, detect changes in oxygen tension, pH, and changes in carbon dioxide. These are two critically important places because they essentially make sure that the important parts of our body are all being perfused adequately. The receptors in the carotid sinus and the carotid body ensure that our brain is being perfused, while the receptors in the aorta are essentially monitoring that our entire system has stable blood pressure and oxygen tension. It's important to remember that the aortic arch transmits its afferent signals via the cranial nerve 10, or the vagus nerve, cranial nerve 10, while the carotid sinus and carotid body receptors deliver their afferent signals through the glossopharyngeal nerve, or the cranial nerve 9. You can remember this by reminding yourself that the vagus nerve, or cranial nerve 10, is the only cranial nerve that leaves the cranium. The rest of the 12 or 11 cranial nerves generally stay in the head and neck region. The vagus nerve is the only cranial nerve that leaves the head and neck region and enters the thoracic and abdominal cavity. Baroreceptors are highly branched nerve fibers that interlock with the elastic fibers of the vessel wall, kind of like vines growing into the bark of a tree. So let's incorporate the physiology of carotid massage, and so we can understand how these baroreceptors work. Essentially, these baroreceptors are going to sense increases in blood pressure at that area in the vessel. So when you massage the carotid artery, we simulate an increase in systemic blood pressure because we're putting external pressure on the receptors. We're increasing the blood pressure within the vessel at that point, resulting in a perceived increase in blood pressure. This increase in wall pressure will cause the baroreceptors to have an increase in stretch, and as such, it'll induce the receptors' ion channels to open and allow an increase in ion influx, resulting in activation of the baroreceptors and a depolarization impulse that travels back to the brain, or the brainstem specifically, through the specific afferent fibers, either the glossopharyngeal or the vagus nerve. This ultimately results in adaptations of the parasympathetic autonomic nervous system or the sympathetic autonomic nervous system. The brainstem senses this transmission from the baroreceptors and causes an activation of the parasympathetic or sympathetic nervous system. So if you have an increase in blood pressure, say by pressing on the carotid sinus in a carotid massage, do you think we would activate the parasympathetic or the sympathetic nervous system? Well, our body is going to respond to a significantly high blood pressure by trying to reduce the high blood pressure, and therefore we would see activation of the parasympathetic autonomic nervous system. We would need to decrease our blood pressure by decreasing the cardiac output or decreasing the systemic vascular resistance. And this is exactly what the parasympathetic autonomic nervous system does. It decreases our cardiac output and our systemic vascular resistance. So really what happens is the neurons in the medulla, as we can see here, the neurons in the medulla go down the vagus nerve, send signals down the vagus nerve to the AV and SA node, causing a reduction in heart rate, causing a decrease in cardiac output. The parasympathetic nerves will increase acetylcholine release in the SA and EV nodes. It'll act on that hyperpolarized, activated cyclic nucleotide gate, the HCN gate on the pacemaker cells. It will decrease the slope of the phase 4 in the pacemaker action potential and result in a slower heart rate and a reduction in cardiac output. This will cause a subsequent or correlative change or reduction in blood pressure as a result of increased pressure sensed at the carotid sinus baroreceptor. The converse happens in settings of hypotension. In settings of hypotension, we have a decrease in stretch of the baroreceptors, and we actually want to have a subsequent increase in cardiac output and systemic vascular resistance to counteract that hypotension. So in this situation, we will actually see a decrease in parasympathetic autonomic tone. So we have a reduction in the parasympathetic outflow, resulting in the SA and AV node increasing their heart rate as a result in order to compensate for hypotension by increasing their cardiac output and systemic vascular resistance. So now let's move on to discuss the chemoreceptors. Chemoreceptors, similarly to the baroreceptors, exist in the carotid body, carotid sinus area, as well as in the aortic arch. These are known as peripheral chemoreceptors because they exist outside of the central nervous system. When we talk about central chemoreceptors, we're talking about ones specific to the central nervous system. Both central and peripheral chemoreceptors respond to changes in carbon dioxide, partial pressure of carbon dioxide, pH, and oxygen tension. But an important point to remember is that oxygen is only sensed in the peripheral chemoreceptors. Central chemoreceptors do not sense oxygen saturation, only in peripheral. So to review, all three of these are sensed in the chemoreceptors both peripherally and centrally, but only peripheral chemoreceptors can respond to oxygen tension. And really the reason is because oxygen has a very difficult time crossing the blood-brain barrier. In fact, carbon dioxide is 20 times better at passing through the blood-brain barrier than oxygen is. So at the end of the day, if the brain needs to be involved in changing respiratory rate significantly, it's better served at measuring carbon dioxide since it's much faster sensed and changes in carbon dioxide are much more rapidly appreciated in the central nervous system. So here we have an image of the blood-brain barrier that the carbon dioxide readily fuses across. It's also important to remember that central chemoreceptors actually measure carbon dioxide indirectly. It measures it indirectly in the form of hydrogen ion concentration. If we remember back to our principle that carbon dioxide plus water makes bicarbonic acid, which is then dissociated into H plus and bicarbonate, we remember that carbon dioxide and the hydrogen ion level are tightly associated with each other. When there are high proton concentrations, we know that there are high carbon dioxide concentrations, and so the central chemoreceptors are actually sensing hydrogen ion concentration as a surrogate for carbon dioxide partial pressure. The enzyme responsible for this is carbonic anhydrase, just as a quick reminder. So when the central chemoreceptors sense an increase in protons or increase in hydrogen ions, will actually signal to the muscles of the diaphragm to blow off carbon dioxide through the phrenic nerve. So when there's high carbon dioxide levels, we see a acidosis, a respiratory acidosis. Our brain perceives that and says, oh man, I need to breathe faster. I need to blow off some of the CO2. It communicates through the phrenic nerve to the muscles of the diaphragm and causes hyperventilation. So that's one of the key mechanisms of the chemoreceptors, the key functions of the chemoreceptors is to measure carbon dioxide levels, measure proton levels in order to manage the respiratory rate in order to keep our acid-base balance within our bloodstream within normal range. So now let's move on to do a board-style question. A patient is brought into the emergency department unresponsive. She is accompanied by an unhelpful man who reports that the patient had used heroin a few hours earlier and was really in a great mood. She then fell asleep and wouldn't wake up. Which of the following is the best description of the compensatory chemoreceptor response in this patient? Alright, so what's going on here? We have a patient who she overdosed on heroin and then fell asleep. So think about heroin. What does heroin do to our respiratory status? Well, as an opioid, it significantly depresses our respiratory drive. So as a result, this patient likely wasn't breathing quickly, wasn't breathing fast enough, she wasn't oxygenating well, and carbon dioxide was building up in her bloodstream. She wasn't adequately blowing off her carbon dioxide. As a result, we see there's an increase in hydrogen ion concentration within the central chemoreceptors, and this would result in an increase in respiratory rate. In order to compensate for this increased hydrogen ion from the carbon dioxide retention. So the answer here is A. Central chemoreceptors will increase the respiratory rate in response to a decrease in pH as a result of hypercarbia, or an increase in CO2. So it's important to remember the key concept here is that carbon dioxide retention in the patient who is respiratory depressed will result in a respiratory acidosis, or an increase in hydrogen ion concentration that is perceived by our central chemoreceptors, with the resulting response of increasing the respiratory rate through communication with the diaphragm via the phrenic nerve in order to blow off the extra carbon dioxide and restore normal acid-base status within the patient.